Now, Punnett square, originally developed by Reginald C. Punnett, it was used as a way of visualizing various crosses, and it aids in problem solving. So the square represents all of the possible combinations in the event. So these are essentially the parents, parent number 2. These are the possible gametes that they could give to their offspring. So if this parent contributes a big G, this parent contributes a big G, the offspring will have that genotype. You would then look at a legend or a chart to tell you this is what 2 big G's code for. Now, if you were to cross something that's round by something that's wrinkled, we have a couple examples listed, or a couple things started here for us. So we're going to ignore this for a moment and take a look at if the question said you were to cross a true breeding round with a true breeding wrinkled pea seed. First thing that you would do is make your chart. Big W codes for round, and it's dominant, and the question would say, in peas, round seeds are dominant over wrinkled seeds, and you would say, so I'm assigning a big W to round, a little w to wrinkled. Little w is what we're using as a letter, or W, sorry, is what we're using as a letter because it's the first letter of the recessive trait. The next step would then be to code the parents. So once I know this, I've read the question, I put down this information. Now the next step here is to put the information into a Punnett square. So first thing that you need to do is draw yourself a Punnett square. Now it could look something like this. You could simply draw a box that looks like this and put the information on the sides and the top. That's your choice, however you'd like to set it up. I like to leave the ends extending out so I can write the information in. Now you pick one parent to go on one side, and you pick the other parent to go on the other side, and so what we will have here is a big W, only one letter per row or column, and this side will be little w's. Now what we're going to do here is exactly like filling in a round robin sheet, finding coordinates on a map. I'm going to take the big W from here and write it in. I'm going to take the little w from the top and write it in, and that's what will be in that square. In the next Punnett square area, we take the big W from the left, the little w from the top, and I write that in. Same idea repeats here. I take the big W from the side, the little w from the top, and I get that. Same idea for the bottom, big W from the side, little w from the top, and the next step would be if the question asks you for the genotypic and phenotypic ratios. So if it asks for the genotypic, geno referring to the letters, so we're looking at how many combinations of big W, little w do we have. In this case, we have 4. So essentially they are all big W, little w. The other thing that you could be asked is the phenotypic ratio. Now, the phenotypic ratio is what they look like. So I would go back to my genotypic ratio and I would say, if they're all big W, little w, I know it needs to be the big W is dominant over the little w, which is recessive. I go back to my chart and I see that big W equals round. And so I would write down here that these are all round. Now, not much of a ratio yet, so you'll notice when you cross a homozygous dominant with a homozygous recessive, you'll end up with all of the offspring being heterozygous. Now, just worked through there for you. Now, if we were to cross the F1 generation, so if we were to say, let's take big W, little w crossed by big W, little w, and it is quite possible that I ask you to go to the F2 generation, we would fill in the Punnett square much the same way. So big W, so one from here, one from here. Here has a little w and a big W. Now, I always like to put the big letters first, so a big W and a little w. Here has a big W, little w. Here has a little and a little w. So just simply looking at where they came from. Then the next question that I could ask would again be, what is the genotypic ratio? And in this case, the F2 generation often provides an actual ratio. So we have 1 big W, to 2 of these, so 2 big W, little w, so 2 of those to 1 little w, little w. So this here is your genotypic ratio. Now, they could ask you also for the phenotypic ratio. So phenotypic ratio is what do they look like. So take a look here. We know that a big W, when we went back to the other slide, said big W equaled round. So we know that we have at least one round. And you might want to start a little tally. Now, when we look at this one, we know it has a big W, so we know it's round, and we know there's 2 of them. So in total, we would have 3 round. Now, we have little w, which refers to wrinkled, so I would write wrinkled, or I could write wrinkled seeds, and there would be 1 wrinkled. So 3 round seeds to 1 wrinkled seed. In this case, it should just be seed. So you can use a tally if you'd like to help you keep track of that. You can just write it in when you take a look at it. If you know it is a monohybrid cross, that means there should only be 4 spots. 1, 2, 3, 4 spots in the Punnett square, so these have to add up to 4. A few things on types of monohybrid crosses. We have a purebred cross. That means, or a pure cross, pardon me, you're taking a true breeding plant, crossing with a true breeding plant, so you're dealing with things that are homozygous. So as you follow along here, you might want to note that those are homozygous. As soon as you're dealing with a hybrid, you're dealing with the F1 generation crossed by the F1 generation, or you're dealing with things that are heterozygous. The other type is also called a test cross. A test cross means a cross between an individual of unknown genotype with an individual that's homozygous recessive, because based on the ratios, we can always tell what will happen. So, if homozygous recessive is known and all we know about the plant is that it has a dominant trait, for example, it has round seeds, but we don't know if it's heterozygous or homozygous, we could cross that pea plant with something that's homozygous recessive. Now, when we go through and take a look at the offspring, if this plant, or this cross, shows every single plant with a big W, little w, now of course when you look at the plant, the genotype is not going to show through, but when you look at it, you probably will see that these are all round. So if they're all round, that means that its genotype would have been 2 big W's. 2 big W's would mean that this is homozygous. Now, if we look at the other scenario, and we crossed, for example, one that was heterozygous with one that was homozygous, we would have 1 big W, little w, to 1 little, which means when you look at the plants, because again, you wouldn't see the genotype on the plants, you would only see that or you would only know that because of what you know about the plant. You would see the phenotype, you would have 1 round plant for every wrinkled plant. Now, basically, you know if it's homozygous dominant like it was in the previous question, you get all the offspring the same. So here, as soon as you get a difference, that's going to tell you that this one had to be heterozygous. So that's a test cross. Now, here's an example or a sample question that you can take a look at. And if you'd like to try this on your own to see if if it makes sense, then you can pause this right now. Otherwise, here's what you would do. Now, I would read through and highlight or underline as I go. So in people, freckles is a dominant trait. Now, first thing I would do is make a key. So we're dealing with freckles. And freckles is a dominant trait. So we could say that non-freckles or non-freckled is the recessive. So I could use a little n and a big N. Now, I use little n simply to represent non-freckled. So if you crossed a person who is homozygous for freckles, knowing the letters that I'm using, homozygous for freckles, homozygous means that they're the same. Everything has to come in a pair. So 2 big N's is homozygous for freckles. Now, the next one is dealing with a person who is homozygous for no freckles. No freckles means little n, so we know they come in pairs, so little n, little n. So there's the one that is homozygous for no freckles. It's asking me what the offspring is going to look like, so essentially what the phenotypic ratio is. So I'm going to put these into a Punnett square. So as you go through the Punnett square, you know that one parent goes to one side. Only one letter per row or column. This parent's going to go to this side. And then I'm simply going to fill them in from the side and the top. And I'll notice that in this example, all of them are big N, little n. Now, when I go back to my chart and I say, what do they actually look like? If they have a big N, I know that they have freckles, so the phenotype here is all freckles. So all of the offspring here would have freckles. Now, part B, if you crossed an individual from the F1 generation, F1 is what you solved for in the previous question, so we know that that was a big N and a little n. With a person who is heterozygous for freckles, heterozygous for freckles in this case refers to big N, little n, so essentially we're crossing the F1 generation together. What would the genotypic and phenotypic ratios be? So, first step to get to your ratios is always to make the Punnett square, because it always gives you those ratios as you go. So big N, little n, big N, little n, and I'm going to fill them in. And then I'm going to count them up. So my genotypic ratio, because it's asking for genotypic and phenotypic, genotypic being the letters, I know I have one big N, I have two big N, little n. Some of you will start to say that there's a pattern here, and I will have one little n, little n. So no matter what the type of monohybrid cross is, if you're crossing two individuals that are heterozygous, you will end up with a 1-to-1 genotypic ratio. Now the phenotypic ratio, I would take a look at this. Both of these are going to code for freckles. So we have 3 freckles to 1 with no freckles. So your answer is 3 out of the 4 offspring would have freckles, or a 3-to-1 ratio of freckles to no freckles. So what I would like you to do next is take a look at, on the home page, I have some sample questions set up for you, and what we will do is go back to the Bio 30 homepage. Just disappeared onto the other screen for a moment. And if you're taking a look in, and again, if you're not sure how to get here, when you hit the Bio 30 button here, genetics, this is under outcome number 2. We started with mitosis and meiosis. Here's the introduction to genetics, a Gregor Mendel PowerPoint that we're taking a look at right now. And then there's monohybrid cross questions. Now when you follow those questions, you're going to take a look at the link that's going to pop up in a Wikispaces. Now what it's set up is some questions at different levels for you to try. So take a look at the level 1 questions, go through those and see if you can work your way up to the 4 different levels. So for example, it gives you some hints on how to go through these, and then you can go through, take a look at the question, try the next one. When you do the answer check, it takes you to a video. The video will play through a step-by-step explanation of what's happening, and I'll show you a picture of that. So when you're ready for level 2, take a look at level 2 monohybrid cross questions. I would say if you do 1 or 2 from each one, you'll be good. Answer check in this case is all in one. So when you take a look at this one, all of the links go to the page that's listed right here. Then you can try the level 3 questions if you'd like to take a look at those. Level 4 is you making up your own. So ideally you should be able to get to level 3. That would be the best version for you. And again, that information is listed on Mrs. Pipke-Painchaud's homepage under genetics, outcome 2, and then monohybrid cross questions. Or you might have been assigned the ones from the duotang. So listen carefully to see what was assigned for this class. Thanks for listening. Bye-bye.